in this video, we're going to focus on transcription. But before we do, I want to review the flow of information between being stored in the nucleus of a cell. So if this is your cell, your DNA is stored in the nucleus in this chromatin format. And this is DNA kind of wrapped around histone proteins. And so if we take a closer look, we're talking about how do we get information from this nucleic acid, deoxyribonucleic acid, to the organism. Now, DNA must remain in the nucleus. Thus, in order to access that information or to do something with it, we need to make a copy of it. And DNA remains in the nucleus, much like we would keep our constitution behind quite a bit of glass, and especially environmentally controlled chamber with specific types of light. Nobody touches it, but most of us have read some part of the Constitution because copies of it have been made. So this DNA can be turned into copies of needed sections, and those needed sections are RNA. And the process of making that RNA is called transcription. This is going to be the focus of today's video. That transcription leads to RNA. RNA is then going to be shipped to a ribosome. This will be covered in the next video of translation. And at a ribosome, we are going to make a polypeptide chain. And that process is called translation. And the polypeptide chain is going to be folded into a final protein. And there's our final gene expression, at least the way we tend to think about it at the high school level. So this polypeptide chain is folded, and that makes your final protein. This whole process is something that we can call gene expression. And it's a simplified way of looking at how you get information from DNA that's stored in the nucleus out to the functional protein that makes our bodies up and creates enzyme that is the enzymes that make us function. So we need to talk about this part of this picture today. All right, so we're going to take a closer look at transcription in RNA. And first, a couple words about RNA. RNA stands for ribonucleic acid. It is a nucleic acid, which means it is made of nucleotides. These nucleotides, though, have ribose as the sugar instead of deoxyribose. And they have another molecular difference, and that is that instead of thymine, they have the nitrogen base uracil. So we'll see A and U matching instead of A and T. RNA, when compared to DNA, is often single-stranded, but not always. It's much shorter or smaller, and it's temporary in nature, so RNA can be disposed of. So it's disposable or temporary. So the copies we make don't have to last forever. And that's actually important because of one of the reasons we don't the DNA doesn't leave the nucleus is because it's so critical that it remain intact. So it stays behind its extra barriers in the nuclear envelope for protection. Two more things. RNA comes in 3+ varieties. And some types of RNA can be catalytic. In other words, they can cause reactions to happen. So if we divide up our RNA into these three variations here, we will focus on 3, but there are more than 3. The 3 variations that are going to play a role in gene expression are going to be the mRNA, which stands for messenger RNA, and its job is to carry the instructions for making proteins. And it has this tendency to be single-stranded, carries a series of nucleotides. And messenger RNA can be anywhere from 100 to 10,000 bases long. And most of the time you're going to find something in the middle of that. 100 is pretty small, and 10,000 is really large, like our dystrophin proteins. So our messenger RNA is going to carry our recipe that carries the instructions for making a protein or a polypeptide chain. And so we're going to say it's kind of like the recipe. Now, the second variation that we're going to talk about is tRNA. tRNA stands for transfer RNA. Transfer RNA starts as a single-stranded RNA, but it ends up folded up into an odd shape, something that looks a little like that, where it forms some double bonds here, and then has 3 nucleotides sticking off the bottom. And its job is that of the chef. It's going to read the recipe. In a sense, it's going to add ingredients in the right order. And that's kind of what a chef does. The ingredients we're talking about are amino acids. So we could say in a more scientific way that tRNAs are responsible for moving and placing correct amino acids. Our last type of RNA is going to be ribosomal RNA. Ribosomal RNA, the picture I'm going to draw is actually of a ribosome. So if this is our ribosome, we'll see that in translation. Made into this ribosome, you're going to see our ribosomal RNA. And the ribosomal RNA is a functional part of the ribosome. And this is where the protein is made. So like any good recipe, it comes to life in the kitchen. Transcription. Transcription is going to be the making of this RNA. The process of making RNA from sections of DNA. And if you think about your DNA existing kind of in this twisted form, parts of that DNA are visible, and there are specific sequences like the Tata box that allow for the pairing of enzymes. So our first step in transcription is going to be what we call initiation. And this is going to be the recognition of a promoter sequence by a series of transcription factors that includes RNA polymerase. So recognition of the promoter, transcription factors, including RNA polymerase bind here. And I should probably be a little bit more specific, and it's the RNA polymerase that's binding to the promoter. The transcription factors mentioned here are going to bind to other sequences in the DNA. So our TA box here is a really, really typical promoter, the Tata box. It's pretty highly conserved as well, although there are some other promoters out there. This is one that you're going to see over and over in this class. Step 2, then, of our RNA transcription is now that we've recognized our promoter sequence, I've filled in the bases on this DNA and opened some of it up. And this would be happening with the help of a RNA polymerase, which I'll draw here in the background. And the RNA polymerase, as it's moving down the DNA, is going to open up the DNA, but it'll close it right behind it. So we have our promoter, and we've already done step 1, in which our RNA polymerase has bound to the promoter and is able to continue or to begin the process of transcription. And it's fairly simple. Step 2 here is going to be called elongation. And this is all about base pairing. So in elongation, RNA polymerase base pairs to what's called the template strand of DNA. We'll come back to this word template in a minute. But our polymerase is going to start pairing. It starts after the promoter. So if our promoter is TA, it's going to start base pairing to our template strand, which is in this picture the lower strand. It's going to pick up after the promoter and it will start base pairing according to complements. Our T is going to pair with A. However, when we get to the A, it's going to pair with U instead, and that's going to continue on from 5' to 3', because if you remember, nucleotides can only be added to the 3' end of a sugar. And that is going to continue until, in step 3, termination occurs. And in termination, RNA polymerase reaches the terminator, and that releases the DNA from the RNA polymerase and ends the transcription. So terminator sequence tends to be a little bit longer than the promoter sequences, and there's a little bit more variation out there. We're going to use one that isn't a real terminator per se, but it works for us and in class, and it's similar to some terminators. It's just a little bit shorter. And that is going to be GGCC. And again, this is a little bit shorter than what we would normally use, but for use in class, it will work to use something that's only 4 bases long. And this causes the release of the DNA. All right, and the new RNA will be let go at that point in time too. So I want to come back to this idea of our template strand. The template strand is what we're going to build our base pair from, or what the polymerase will be doing its base pairing from. And it's important to recognize that when you read these sequences, they are complementary and not identical. So the letters and the order of the letters is different on the top strand versus the bottom strand. If we turned our paper around, it would be the left strand and the right strand. And even if we were to read backwards from the same point, it is not going to be the same sequence of nucleotides. And that will become apparent why that's so important when we get to translation. But for now, we need to recognize that we're going to base pair to the template strand because it's going to give us RNA that is most similar to our coding strand. And the coding strand is what contains the information we need for our RNA. So our coding strand here contains our needed information. But in order to get the coding strand, we will have to base pair to the template strand, right? So once our RNA polymerase has reached the termination sequence and has let go of the RNA and the DNA, there's one step left to go in eukaryotic transcription, and that is RNA editing. In the image here, I have blue and red DNA. So this is going to be called our pre-mRNA. And so this is not ready to leave the nucleus yet. It's not done. It needs to be edited. And so in eukaryotes, we have 2 parts to our RNA transcript here, pre-mRNA. And that is that we have exons and we have introns. And the exons are what we're going to end up keeping. And I did them in blue. And our introns are going to become garbage, and we're going to get rid of them. There are a number of different reasons that we think introns might exist in exons. Introns may play roles in regulating other genes further down, but when we go to make an RNA molecule, we don't need these introns anymore. They're involved in regulating some other gene further down the genome. They may be ways of allowing us to take this pre-mRNA and make several different proteins out of it, depending on how we put these pieces back together. So RNA editing in a really simplified version is going to cut up the blue and the red and paste the blue back together. It's actually a little bit more complicated than just going through and pasting it together. It happens with the help of a spliceosome. And the spliceosome is something that's going to come in and identify sequences on either end of the exon, and it's going to work at this point in time to kind of loop that or that intron out, and it's going to then cut off the intron so that we now have this loop of DNA that was the intron, and we have our exon that's now in one piece. And that's going to happen all the way along. So if we were to see this kind of zoomed out, we'd have all of our all of our exons here, and in between we'd have the spliceosomes kind of looping out the introns that are no longer needed. And so you use spliceosome for each one of these. All right, now that doesn't all happen at once, but you get the gist of what is happening. So this is our spliceosome. And that spliceosome is doing the work of cutting out the introns and pasting our exons back together. And so when we get down here, our spliceosome is done working, we have this strand of DNA. It now is correctly coding without the introns. It isn't quite done yet. It needs to have a cap and a tail added. And the cap and the tail play roles in regulating how this is getting out of the nucleus and how it's going to connect with the ribosome, and also protecting the RNA transcript itself from some degradation. Now that we have a cap, a tail, and our edited RNA, we can now say that this is a finished messenger RNA molecule and is ready to be used.